today I've got with me Professor Frank Martin, who's my old boss from 2005, who taught me everything there is to know about research, from barely knowing anything at all to knowing a little bit about something. And so Frank is professor at University of Central Lancashire, and is a professor of biosciences. Is known as a toxicologist, probably one of the top toxicologists in the world, I would say, and has over 200 publications in some pretty big journals, including Nature and The Lancet, I believe. Is there any other good ones out there?
A few in my particular fields, yes. Toxicology, cancer research, carcinogenesis, those kinds
you
of
name
things.
it, you've done it.
I've done it.
And actually, Frank was actually pivotal in my research in endometriosis and endometrial cancer. So it's quite a wide area that you basically do, Frank. And
it is interesting because what I've discovered through the years is that a lot of the techniques that you might apply to one particular problem are translatable to other problems. And of course, as we want to progress in our knowledge, we take advantage of findings in one particular area and apply them to new areas.
Yeah. So what I wanted to talk to you about, because we obviously had many coffees together, and Frank, we basically cured cancer between us in our heads anyway. But but we used to
give it a good go.
We gave it a good go. Just an hour of coffee every week. I recommend it to anybody out there. But to have that discussion at that time, and I wish I'd recorded those conversations then, because we used to come out with some really good stuff. I thought myself, but but what I really want to talk to you about is how health has progressed and where we're at the moment and where we're going in the future, and then basically to move into your areas of diagnostic medicine and where we go from there. Let's just see where we go. So
Sure.
So what do you think are the major health problems at the moment here for our population and for the population of the world at the moment?
Well, the major health problems at the moment is, of course, incidence prevalence of cancer is increasing as we're aging. Also with aging, neurodegenerative diseases are increasing in their prevalence. And also, as we're becoming more affluent, maybe in the developed world, more sedentary maybe, obesity is also emerging. And that has knock-on consequences in terms of increases in diabetes. And then there's a relationship between diabetes and these other aforementioned diseases, cancer and neurodegenerative disease.
Yeah.
So understanding that relationship is going to be a critical area for us in the future.
And is it? Could we improve our lifestyles to make things better, or is it purely obesity, or are there other factors that might be affecting us? Not just obesity, but other external environmental toxins or whatever.
Well, it depends on how much we're programmed to, what is our life expectancy and what's the optimum life expectancy in the human population. We hear a lot of reports about what's good for you, what's bad for you. Of course, moderation in diet is important. Moderation in terms of alcohol intake is important. Smoking has its disadvantages, its side effects. So those controlling and exposure to sun, controlling those factors is important. Again, exercise, there's balance with exercise. How much exercise do you? So marrying those things with our modern lifestyle, what does that give us? And that's the major question in terms of.
What sort of foods? We hear every week. There's some foods. There's these seeds, those seeds. There's something else is good for you. Then the week after, it's not good for you. What, in your opinion, is good for you, and what isn't good for you?
It's without any doubt the major beneficial foods are it's a healthy intake of fresh fruit and vegetables,
right,
and cruciferous vegetables, broccoli, are highlighted, and I do believe that the evidence for those having a compelling beneficial effect is there. We have the French paradox, of course, where people in south of France have a lower incidence of cardiovascular disease and cancer, and that is associated with intake of red tomato-based foods.
Yeah.
So we then highlight things such as lycopene as being beneficial. Again, with the French paradox, we've also highlighted agents such as resveratrol. Resveratrol is a component of skin of grapes, red grapes, and that's where moderate intake of alcohol has sometimes been touted as being beneficial or being having a good being good for you. And other things, phytochemicals such as quercetin, which we see in nuts, we see they appear in onions, we consume in a typical diet milligram amounts of these. The evidence is that they have a beneficial effect. So such foods have a beneficial effect. Then you have what is bad for you. That's processed meats. Right. The evidence that is now there, the processed meats.
So avoid McDonald's.
I wouldn't go so far as that. I wouldn't name, but I would avoid anything that has processed meat and regular intake.
Right.
Red meat also. I mean, of course, you need. I mean, it's again finding a balance because of course, healthy diet will have red meat, but how much you might eat consistently over time. That's the major question. And I'm not sure we quite understand that relationship yet. And of course, you do always have to understand all of these things are on the complex human being, and human being is an extremely complex entity to do research on. We're genetically outbred, which means we respond very differently. We have very different factors in our respective lives. Whether we take exercise, what kind of foods we consume, and the combinations of these things will have modifying effects on each other. So understanding that in humans is very difficult.
And I think we have that individuality, don't we? That's the other problem, that we all respond very slightly differently to each other, and we're not cars at the end of the day.
Absolutely.
And I was going to talk to you about vitamins and supplementation. I'm not a huge fan of supplementation. I don't know where. I know there's a big drive, certainly in pregnancy, to take vitamins and things. I'm not sure whether it's a good or a bad thing. And I know folic acid and iron are definitely worth supplementing to reduce spina bifida. But what do you feel are the role of vitamins in somebody who's got a relatively good diet?
I think the role is at best minimal.
Right.
If with someone who has a good healthy diet.
Yeah.
I can understand the role of iron and folic acid and taking that during pregnancy.
Yeah.
I tend to distinguish those, differentiate those from vitamins per se.
Yeah.
The evidence for taking vitamins as being beneficial for you, as far as I'm aware, isn't there. It's never been proven.
Yeah.
And I would expect that, given the amount of research that's been done over the last number of decades, if there was a compelling benefit, this would be observed.
Okay. And things like, fish oils and things like that, for brain development, is there evidence there?
Again, I would say a typical healthy diet would provide you with sufficient dietary requirements.
Okay.
You also have to remember with coming back to vitamins. The evidence from vitamins being beneficial comes from populations that consumed healthy diet of fresh fruit and vegetables.
Yeah.
So people began to look for what would be the protective factors. These agents are complex, within a very complex milieu of within healthy fresh fruit and vegetables.
Yeah.
And how in that complex milieu, those agents act as very good antioxidants. They're protective.
Yeah.
When you take these things out and put them in a very different environment, it's not absolutely certain that they're going to be protective.
Right.
So that they retain those protective properties.
Right.
So having something in a tablet form isn't necessarily better.
Advantageous. Okay.
Advantageous.
Okay, that's interesting. So, and other, I mean, we talked briefly on fish oil. Fish oil seems to be the latest thing at the moment, and a lot of my patients take fish oils. Is there any? Could you be causing detriment in taking supplementation or extra fish oils? Is there a way? Can you overdose on these things, or
you can overdose? Maybe possibly the detriment, the way these things might be detrimental, would be when they are in isolation.
Yeah.
That they're not conferring the beneficial properties that should be. That they become, rather than antioxidant, they become pro-oxidant. So
Right.
they become damaging.
Right. Okay.
And sometimes you find a classic example of phytochemicals such as quercetin within the nuts or within onions that you find these things. They are very good antioxidant, protective.
Right.
They've been shown by many scientists even since the 1930s to improve vasculature.
Mm-hmm.
But you take these things out in isolation, rather than being protective, they're actually mutagenic.
Right.
In isolation, they're mutagenic. So you put them into a short-term toxicology and mutagenicity assay in the laboratory. They wouldn't. We consume them all the time, and the irony is that they were discovered by a chemist to become a pharmaceutical.
Yeah.
They wouldn't pass any toxicology tests that fall the first hurdle because
Right.
they're mutagenic. So that's
Okay.
the complexity with these agents.
So they almost have to work in the environment they're in
in the environment they're naturally in
to be effective.
To be that would be my guess.
Yeah. Okay. And when we talk, you mentioned to me once about vitamin C in smokers, and a study where they looked at smokers and giving them vitamin C.
This was beta carotene.
Right.
So they gave smokers beta carotene, which was in all the toxicology and the laboratory tests, it was an antioxidant and that was protective. And yet, when it was given to smokers, rather than reducing risk, there was evidence that it might even be elevating risk in these individuals.
Right.
So
if you're a smoker, don't have beta carotene, is what you're saying.
Well,
I'll have to remember that one, Frank. Okay, and so where do you think these rises in breast cancer and in estrogen-related cancer, is that really obesity that's causing that, or is there some other factors that you think may be causing?
There is going to be a complex interplay. If you take breast cancer isn't just one disease.
Yeah.
Especially with genetic platforms now, we're discovering that it could be a range of different diseases.
Yeah.
Majority of breast cancers are what we call sporadic.
Yeah.
And when you look at the incidence worldwide, even now, there is a lower incidence in some countries and a higher incidence in other countries, such as Northwestern Europe and in the US.
Right.
Populations that migrate from these areas of low risk to these areas of high risk, they acquire the same level of risk as the overall population. That points to environment in some fashion or lifestyle. Now you can point then to particular things in our diet, especially the Western diet, and the way we prepare food. We might dry heat food.
Yeah.
This may generate products of protein pyrolysis, which are aromatic amines, which might be mutagenic, carcinogenic, and also obesity as a driver.
Yeah.
And there must be a complex interplay between those things, because obesity will be a pro-estrogenic condition, and certainly that is associated, maybe not so much with breast cancer, but very much so with cancer of the womb, cancer of the endometrial cancer. Right.
And is that bordering on the epigenetic mechanisms that may be out there as well, or is that?
Well, again, this is a whole new era that we're entering. So we've had the Human Genome Project, and that's the code of human life, code of life, and a lot of work has been done on that. But we've increasingly discovered that not only is that code important, but how that code is expressed is important.
Yeah.
And that's really the critical thing, and that takes us into the era of epigenetics, and towards what we might call the Human Epigenome Project.
Right. Okay.
And that expression pattern is what's exciting about that expression pattern is that it's inducible by environmental factors.
Yeah.
So things that might be bad for you or good for you might modify the expression.
Right.
It's also importantly, it's reversible.
Yeah.
So if you identify especially something that's a bad epigenetic alteration, there is potential that we can look at ways of reversing this. So you can't reverse a mutation, but you can actually reverse an epigenetic alteration.
So you basically reverse the way the
expression is occurring.
Okay, in the cell.
In particular, so if you have activation of genes that you don't want activated, or you have silencing of genes that you don't want silenced, you can reverse that.
Okay.
And finally, what we've discovered is it's transgenerational. And that takes us into a whole new era, because the old saying was, when if you wanted to be an athlete, you'd choose your parents to be Olympic athletes. If you wanted to be a great academic, you'd choose your parents maybe to be Nobel Prize winners.
Yeah.
Now, what that tells us, not only do you choose your parents on based on what they are and their genetic makeup, but you also have to make sure that they live correctly and expose themselves to correct exposures before they conceive you, because those alterations those exposures may induce epigenetic alterations, which can then be heritable. They can be passed on to the next generation,
right.
And that's where it becomes extremely exciting.
So even 6 months or a year before they even conceive, or even longer.
Even longer.
Right.
Even longer. There is a very good chance that this much longer. This could be years, and not only that, but once these epigenetic alterations are induced, there is some evidence, it's preliminary at the moment, that these alterations could go across multiple generations.
So delaying your age of conception, so those kids born, from people who are in their 20s to those who are in their late 30s, could there be differences?
Well, that's something I've thought about and something I've considered, especially, we always think of the mother as being the most important,
yeah,
and what things that might modify the mother, but it takes two to tango.
Yeah.
Because the father is involved too,
yeah,
and he contributes 50% of the genetic makeup too, and there may be even evidence that males are more susceptible to these epigenetic alterations than even females.
Yeah,
and that's a field that's beginning to emerge, and that may be associated with perceived or apparent reductions in male fertility that have occurred since the early 1970s. What's controlling? What's behind that? So
it'd be interesting to see the athletes that go to the Olympics at Rio this year, how old their parents were when they conceived them. I'll be quite interested to.
Might be a study.
It might be a study.
The problem is, how would you set that up? And tracking these people and
you basically ask them, how old are your parents when they conceived and where do they live, and were they vegetarians or whatever. Just a simple study for.
Yeah,
we could do it for the British Olympic team, perhaps.
We could have a look at that.
Shall we just give them a ring? We'll see if we can. Because it'd be interesting. I'll be interested because I had my first daughter aged 27. And that was only a couple of years earlier. I was drinking quite heavily because I was at medical school. So I'm a bit concerned that I passed something on from the alcohol side and my more hedonistic lifestyle that I had then to what my very sedate lifestyle I have now.
Of course, but then we have to think about we're just at the very beginning of this journey of understanding epigenetics.
Yeah. And
there may even be evidence that you may have even conferred protective factors to your daughter against alcohol intake.
Well, that's
so right. It might be.
Well, I think there's always a yin and yang. When you try and it's all about balance. I think, and sometimes you push things too far one way, and then you lose the benefits in another place. I think it's trying to find that right balance. I totally agree. So, where do you think health's going in the future then, Frank? Do you think we're going to see more cancer? Do you think we're going to see more?
We are going to see more cancer, and how we deal with that will be one of the major challenges, economic and in terms of looking after people.
Yeah.
And I think certainly in the medium term, we're not going to cure cancer. I think in the medium term, we are going to look increasingly towards disease management.
Yeah.
I think we are going to enter in some fashion the era of personalised medicine, identifying people who can respond better to particular medications. You see that with certain brain tumours already. People will respond to particular cancer treatments based on expression of DNA repair enzymes. So that will be certainly that would be. We are also in this area at the beginning of a very exciting journey in the re-emergence of immunotherapy.
Right.
And where that takes us is going to be, that is, if I was to bet on anything, I would say if we are looking to cure cancer, developing immunotherapy will be the way we're going to do it and achieve that. I don't think with conventional therapy, medication, we can do it. Cancer is too complex a condition, and it's evolved over. If you take cancer as a disease, it's evolved from maybe your teens to when you express the disease as an invasive form.
Yeah.
And that may be 20, 30, 40 years. And in that period, all kinds of mutations, all kinds of epigenetic alterations have occurred.
Right.
Not just in the cells that expanded in this autonomous fashion to become the disease, but also in what surrounds these cancer cells and how that surrounding connective tissue may support and nurture the cancer.
Yeah.
And so being able to cure disease in our conventional fashion, I don't think we're going to do. I think we're going to manage disease.
Right.
But where immunotherapy takes us is another question.
So it's when you talk about immunotherapy, what do you mean by immunotherapy?
That is activating your own immune system to identify the disease.
Right.
And what that means is if you can find a means of activating your immune system because all of us will develop cancers.
Yeah.
And these cancers, many of them will just regress and die away.
Yeah.
Some will just remain latent and do nothing.
Right.
Why do a small number progress to invasive disease?
Right.
That's the major question. And why do they obtain the ability to bypass any surveillance mechanism within the body? They grow autonomously.
Yeah.
Our immune system can't handle them. They can't make them. So finding a way of helping or assisting our immune system to target these cancers, not only will it target the primary disease, but it'll also, because it's been activated, target the secondaries if and when they arise.
Right. So you could, it's almost like a cancer vaccine.
Essentially, yes.
Right. That's interesting.
So that's, for me, that is it. I think there have been case examples that have come out of ASCO even this year of incredible results. Certainly in the area, there have been trials on immunotherapies for skin cancer, for melanoma,
right.
And these are in patients who had very little hope.
Right.
Conventional medicine had failed them, and these people have extremely good survival.
Right.
So it's pointing to something quite new. Now, immunotherapy, and this has been an area that's been around for a long, long time, but it's re-emerged as people have developed.
That's a big candidate for the future, yeah.
I think so, yes.
And so diagnosing cancers early is very important, is it not? So, and I know you've been, look, I'm going to talk about infrared microspectroscopy, and every time I mention this so I've only had a very brief glance with infrared microspectroscopy working in your lab, and trying to explain that to even medical people they don't really get it. And and this is how I started with you originally, Frank, is when I asked you to explain science to me, you started at this level and we worked way down. I said, look, just start at the bottom floor and work your way up. So from the bottom floor, what's infrared microspectroscopy?
Okay, what infrared microspectroscopy or spectrochemical analysis is, and spectro is spectrum, chemical is chemical analysis. We exploit the fact that a portion of the electromagnetic spectrum, i.e. you could say light, interacts with chemical bonds in biological substances.
Right.
And these chemical bonds, these biological substances can be tissue sections, they can be cytology, i.e. cells, or they can be biofluids. The interaction generates a vibration which can be detected. And this can be detected as a spectrum. Right. And that spectrum is a fingerprint of that biological material that you've analysed. So you've shone a light on biological material. There are different ways we can do that. And how the light interacts with the chemical bond induces a vibration in the chemical bond. How that is detected or sensed generates a spectrum. And that spectrum, as I said, is a fingerprint.
Right.
And we can then obtain many spectra, several in typical, we can handle thousands, hundreds of thousands of spectra in typical analyses. And we can then use different computational methodologies, not just to look at differences, but to visualize those spectra, which are data. You can visualize data in space. And what we can do is visualize data in a fashion so that we can identify which samples are similar to each other, and which samples are dissimilar.
Right.
And those samples that are similar to each other co-cluster in this space, and those samples that are dissimilar segregate away. So we can say, well, our cluster of normal samples can be here, and our cluster of abnormal samples can be here.
Right.
And that's important because based on that computational methodology, we can build up what we call objective classification methodologies.
Right.
And why is this important? Because, again, with increasing population, with increasing need to diagnose in an aging population, we need new methods of objectively screening and diagnosing.
Yeah.
Objectively screening because, of course, mistakes are made in terms of specificity, lack of specificity, and there may be a number of reasons for that. Even consultant pathologists will disagree on a diagnosis.
Yeah, absolutely.
And then understanding what you do with that disease. Can you characterize the diagnosis? Can you, in brain tumors, can you characterize it as a glioma or meningioma, or is it a metastasis from an unknown primary tumor that hasn't been diagnosed?
Yeah.
So that's infrared spectroscopy in a small nutshell.
So, and you can test any tissue, any blood.
What is exciting about this new approach is that it is reagent-free, and it can be applied to any biological sample.
Right. That's interesting. And what sort of conditions have you looked at so far? Right.
We have applied this to a range of questions. First of all, to look at cell lineage within a range of different tissues. So we've looked at cell lineage within human skin, in the gastrointestinal tract, in the cornea of the eye, in breast tissue, in ovarian, and endometrium.
Yeah.
We have a very nice paper under review for endometrium currently. We've then applied it in terms of understanding cancer within these tissues. So we've applied that to prostate, we've applied it again to endometrium, we've applied it to breast, brain, different brain cancers. So we've applied it to quite a range of cancers in that area. We've applied it, we've done some very nice work on cytology, especially cervical cytology.
Yeah.
We've done PAPs, absolutely. And what was exciting about this, we did the world's first piece of research where we combined the spectrochemical analyses with underlying histology. And we were able to show that underlying histology picked up undetectable cancers. So when we corrected for this, we found our spectrochemical analyses to be 100% sensitive and specific to diagnosis, even for low-grade disease. Based on that, we could do predictive models for low-grade disease. So when we had a woman that come in with low-grade cervical cytology, we set up again, it was the first piece of work in this area where we could identify which women remained the same, which women would regress, and which women would, the small number of women who would progress to disease.
Right.
And that's really critical because no one has a dichotomous marker of disease progression in low-grade cytology.
That's right. It's always a problem for us because if we get low grade,
what do you do with it?
Yeah, we sometimes sit treats, we sometimes sit tight, and treatment isn't without its risk for future pregnancies.
Absolutely.
So it is an important.
And we've applied this also, to biofluids. So we've done a lot of one of my biggest projects is in biofluids. We have quite a big piece of work looking at neurodegenerative disease, comparing biofluids from Alzheimer's versus Parkinson's disease versus a control cohort,
right.
And Lewy body with dementia with Lewy bodies.
Yeah.
We have done it for ovarian. Certain cancers, I believe, will work in biofluid areas. So ovarian cancer, we have a very high sensitivity and specificity. So that's going to be exciting for us.
Absolutely.
So ovarian cancer is a very insidious disease. If you can diagnose early, you can actually treat.
Yeah. That's right.
Readily treat. It's because these cancers present at a later point. That's why they're so hard to treat.
Absolutely.
Very often.
I mean, that's the problem with ovarian. It comes so late, and the stage 3, 4, and the outcomes are so poor. And when you do get that stage 1, it's curable. And
absolutely.
It's because it's just in the ovary. And that makes it so much easier surgery and chemotherapy wise later.
Spectrochemical analysis, I think, is what I've tried to do with all my research in this area is interface it with current clinical practice, and I've started off so we can apply this to with archival tissue blocks. Typically, any pathology lab will have.
Yeah.
We can, apply to frozen tissue, conventionally collected cytology, and biofluids. It's reagent free, but it's also very low cost. Once you have an instrument, which is not expensive at all in context of what instruments can cost, technology costs these days, the running cost is not dissimilar to current practice. In fact, it's probably cheaper.
Yeah.
And that's where I think my sort of analogy always is that we've used for X number 100 years a microscope, which it has an optical eyepiece, and that has allowed us to look into the microverse. Now, we use telescopes. Galileo used the telescopes looking, into the sky, and of course in the 1950s we developed X-ray microscopes, X-ray telescopes.
Yeah.
And that gives us additional information we couldn't see. And likewise, we're developing these X-ray microscopes that allow us to objectively look at the microverse of the cell, to look at for alterations that we hitherto could not identify.
Right. That's amazing. It is. Look, I think that the future of that. I mean, when I first came across infrared microspectroscopy with you in 2005, it was difficult to understand, but we've progressed an awful long way in 10 years, I think. And in what you're doing, and it's just getting more and more important as time's going on. How soon do you think this will come into play for the Joe Blogs on the street?
I think in certain areas it will come into play quicker than in other areas. I think in terms of oncology, things move slowly in oncology because you cannot have one cannot make a mistake in oncology. If you miss a cancer, you've got a problem.
Yeah.
So changing current practice, in my mind, will take 10 to 15 years at least.
Yeah.
Some of the instrumentation is laboratory. And certainly there are people looking at it already, means of converting these technologies into a point of care clinical setting technology.
Right.
So there is work in that direction. But even so, validation of these methods, validation in parallel with histology, will take, because you need follow-up, putting in place that kind of robust
structure.
Absolutely.
Yeah.
Takes and it's very, and one of the difficulties is that it's such an interdisciplinary problem.
Yeah.
So you need this crosstalk between the clinician, the scientist, in the science, the person who understands the biological problem, the person who understands the physics of the sensor that you're going to use, and also the bioinformatics person, the computational person.
Yeah.
So that's a big team. Now, ultimately, once you have the framework in place,
yeah,
you don't. It's a technical problem, a technical job. It's a handle turning exercise. You have. I mean, I used the sort of the red green light for this. You have a sensor that you put on a biological sample. You zap it with the infrared. You get a spectrum, and then you have a computational algorithm that will then bin that spectrum depending on whether it's within the normal reference range or whether it's outside. If it's outside, you get a red light. If it's within, you get a green light. You just progress. Then you investigate these samples that are outside a little further.
Right. So it's almost a screening process, and then that becomes a diagnostic process.
And then you can take that further for diagnosis.
Right. Can you do staging of endometriosis? The reason I'm asking this, obviously, is because I'm waiting for this bloody paper.
Sure.
Do you think you could? So we've done this work on
the endometriosis work was one of the most compelling pieces of work we did. We did this a few years back, where we obtained eutopic versus ectopic tissue.
Yeah. And
what was most compelling for me was, first of all, we could segregate out, independent of any inter-individual variability, we could separate out the diagnostic features of endometriosis.
Right.
Not only that, but we could actually show in eutopic endometrium that there was a difference in those women who had endometriotic lesions.
Yeah.
Now, in the subsequent work which we've done, which we have to publish, the student who did the work has got her master's. She went on to Cambridge after that. We can't separate different
stages.
Absolutely.
Because that was the thing, was that the line of the womb, the endometrium, was always going to be different for those with endometriosis. That's what we always thought.
Yes.
And it's interesting to see that we can show
I believe that's the case.
Yeah, absolutely. And moving away from infrared mass spectroscopy, why do you think we're getting older, Frank? I've always wanted to ask you this. Why are we? Is it just food? Is it other things? What's the process of aging?
Well, in the Western world, we've been very lucky. We've had, era of peacetime. We've had the era of antibiotics. We also have had increase in education. We are educated, so that means we know how to look after ourselves better.
Yeah.
And there's less physical work, and we don't work as physically as our grandparents would have worked. You don't have 12-year-old kids going down to, mine shafts anymore.
Yeah.
You don't and of course nutrition is much better. We understand nutrition, even though we don't think we do. We do understand nutrition far better than we did.
And do you think our kids will live forever? Or live? Are we going to come to a generation in the next 4 or 5 generations where people are going to live for a bloody long time? Or
it depends where regenerative medicine will take us. Because we need spare parts. It'd be like do we need to grow a new eye? Because we're getting macular degeneration in the old eye. Can we move our thoughts from that's an even more complex interdisciplinary topic, because that not only takes you from social sciences, the psychology and the biology. It's a very
because if I was to live longer, I'd like to live a good life. I wouldn't like to live a.
Well, that's one of the major questions. I mean, are we living longer? But are we living longer sick?
Yeah.
And that's again, that's of course we have plenty of medication. We can take plenty of medication, and how that maybe reduces the onset of a particular pathology or disease is a good question. But
so one of my interests is I really want to look into aging, and and trying to either reverse it or stop aging. At a certain age, using a concoction of chemicals. And that's a real interest of mine. But my worry is that is to give it out to the population, because if you give it out to the population, then there's huge ethical issues with having a population that don't age and the world becoming over-inhabited. So,
And of course, your lifestyle might change, and certain habits might change, and certain. But I don't think we're that. I don't think we're close.
Well,
I think we're more than 4 or 5 generations away from that.
Well, Frank, if you still see me looking the same age as I am now today, then in about 20 years' time, you know I've cracked it.
Absolutely.
But Frank, look, it's always a pleasure to speak to you, and I wish I had more time. And every time I talk to you, I wish I was back in England, working more closely with you research-wise. And but Australia still has a draw for me, partly because of the weather, but I'm sure we'll continue to do a lot of work together in the short term. But in the long term, I do see myself back here working very closely alongside you, because I think some of the stuff you're doing is absolutely amazing. And I still do, and I don't get embarrassed when I say that, I do think you are probably the greatest scientist I've ever met. And one day, I'm sure everyone else will appreciate that too. And I really appreciate you spending the time with me today, because I know you're so busy. So thanks again, Frank.
It's a pleasure. It's a pleasure.
Absolute pleasure. Cheers, Frank. Cheers. See you.